Going to be our first biomolecule that we're going to be looking at. And carbohydrates are classified or defined as polyhydroxyl aldehydes or ketones, and or anything that can be broken down to form these compounds, right? So generally when you eat carbohydrates or sugars, well, not sugars necessarily, but carbohydrates, they're in a long polymer chain. Those can be broken down into polyhydroxyl aldehydes or ketone. So polyhydroxyl, what do you guys think that means? Many, right? So this is going to have many different hydroxyl groups, right? And basically at every spot along a carbohydrates, these, at any spot along the carbon chain of a carbohydrate, we have a hydroxyl group. And so the ones that can be broken down, again, these are going to be polymers of single sugars or carbs. And how we define a carbohydrate in terms of a, if we were to draw a picture of what a carbohydrate looks like, an example of one is they're represented by these structures that look like this. And at every spot along the carbon chain, we have a hydrogen and a hydroxyl group. So here are all the different hydroxyl groups that we have on this carbohydrate. And just to preface, these can be in a linear structure or a ring structure. And we have a couple definitions for different carbons on this chain. The anomeric carbon is the carbonyl carbon. It's called anomeric for what it functions as in the ring structure of the carbohydrate. And so that's going to be this carbon right here. And then we also have another carbon called the penultimate carbon. And that's going to be this carbon right there. And the penultimate carbon is ultimately going to determine if the sugar is one enantiomer or another of each other. So back when we talked about chirality and stereochemistry, that is going to come into play when we talk about carbohydrates and amino acids. Right? And if we're looking at this structure, each intersection represents a carbon, and OHs are drawn. So what I mean by that is that at each spot here, those all represent carbons. All right, so each intersection represents a carbon. And whether we have a aldehyde or a ketone, that'll change the number of carbons. So this is just as OHs are explicitly drawn. I don't think I necessarily needed to write that because you can see that I drew all the OHs on this carbon. Maya? Penultimate carbon. Penultimate carbon. And so these are some of the basic definitions for an aldehyde or a ketone. And some of the consequences of this definition is that for a carbohydrate to exist, it needs to be at least 3 carbons long. Here? are drawn explicitly. So we draw all the OHs and the hydrogens explicitly in this structure right here. And I'll let you guys know these are called Fisher projections. Is what they're called. Coffee. I don't know. I feel like there's no good way, because then that's. It has to be at least 3 carbons long, and so we'll go through, the next slide will be an example of and it should start to become apparent why that's the case. Okay, so here we have a molecule. Determine if it is a carbohydrate or not. Right, so what was a carbohydrate again? Ketone or aldehyde, and it has to be polyhydroxyl, right? It has to have multiple OH groups on it. So is this a carbohydrate? Is this an aldehyde or a ketone? Where's the aldehyde group? At the top right there, right? This, and how do you know it's an aldehyde or a ketone? Double bond O and right next to an H, that means it's an aldehyde, right? So this is an aldehyde. So it's an aldehyde, so that's good. And then the other definition, or the other requirement for the definition was it had to be polyhydroxyl. Is this polyhydroxyl? Yes. Yep, because we have one OH here. Oops, I meant to do a different color. One OH here and one OH group there, right? So multiple OH groups. So yes, this is a carbohydrate. And how many carbons are in this carbohydrate? 3, right? Okay, what about this molecule here? So why don't you guys take a second and just determine if this is a carbohydrate or not. Is this a carbohydrate? Yes. Why? Who says yes and who says no? You say no? Why do you say no? Yep, this isn't a ketone or an aldehyde. We have no carbonyl here, right? Which means there's no carbonyl. So it's not a ketone or aldehyde. There's no ketone or aldehyde, which means no, this is not a carbohydrate or a saccharide, monosaccharide, polysaccharide, trisaccharide. This is actually a molecule called glycerol, which is responsible for a specific type of lipid, which are called triglycerides, right? So this is a molecule that is part of triglycerides. We'll talk about that when we get to lipids. It is polyhydroxyl, right, because it has multiple OH groups there, but it is not an aldehyde or a ketone, so it's not considered a carbohydrate. Okay, what about this molecule? Have a carbonyl. So this means it's probably going to be an aldehyde or ketone. And which one is it going to be? Is it going to be an aldehyde or a ketone? Ketone, right? Because an aldehyde looks like that, and then a ketone looks like this, right? Aldehyde is the carbonyl carbon is directly bonded to the hydrogen, where a ketone, it's bonded to another R group, right? So this is what we have here. So this is going to be a ketone. And is it polyhydroxyl? Yep, because we have an OH group here and an OH group here. So it is polyhydroxyl. So this is a carbohydrate. Right, and one thing to notice here, because we had one aldehyde carbohydrate, and this is a ketone carbohydrate, is that the ketone carbohydrates have that extra CH2OH going up, and aldehydes only have the H going up. Right, so that'll, whenever you see an extra CH2OH group, that means you're going to have a ketone carbohydrate. And we'll talk about how those are classified. Right, so what I'm saying here is this one has the H going up. So that is an aldehyde carbohydrate, and this molecule has a CH2OH going up. And so those are going to be indicative of carbohydrates. Okay, any questions on identifying carbohydrates? Yeah. Yeah. This one here? Okay. We're good to go on? All right, so carbohydrates are synthesized in photosynthesis in plants. And so carbohydrates are used for producing energy in biological systems. Biological systems. And you guys have probably studied at some level photosynthesis. And so what does photosynthesis do at a very basic form? If you were saying a plant was photosynthesizing, what would you say it's doing? Creates energy from sunlight, right? And so if we look at this chemical reaction here, we see this energy value right here. This is going to be light that goes into this reaction. And we start with some carbon dioxide and water and make carbohydrates from that. And so we're starting with reactants that are relatively low in energy. So these reactants are low in energy, and we create products. This is a carbohydrate, and oxygen is a byproduct. We create products that are high in energy. These are used for energy in the plant. They have to take in energy from the sun to synthesize these carbohydrates, and then they can later be used in some sort of metabolic pathway for metabolism to then start. And so these products were the carbs and sugars to then go back down, break up these carbohydrates and now in this case of this reaction, we have a largely exothermic reaction. Right, so delta H is going down in energy, and that extra energy can be used for other biological pathways or side reactions. All right, so the idea with photosynthesis is we take energy from the sun, or we don't, plants do. They take energy from the sun to create these molecules that have a lot of energy stored in the chemical bonds. When we go through a metabolic pathway, we can use these molecules for energy by breaking them down into lower energy products from there, most likely carbon dioxide and water as a byproduct of that process, to then use that energy for other chemical pathways, other biochemical pathways. And so that can be used for making a protein, for example, right? We said that the making an amide bond took energy because it's an unfavorable reaction. We can use this energy to then synthesize proteins inside the plants. And so carbohydrates are used as immediate energy reserves in biological systems. If we need energy storage for a lot later, does anyone know what biomolecule we use for that? Fats, right? Lipids and fats. In a lot of times, this energy is stored in what we call glycosidic bonds. So these are bonds that hold different carbohydrates together in the ring structure, and we use these glycosidic bonds to store energy. And so when we have bread, for example, we have a polymer of carbohydrates. We digest the carbohydrates, we break these glycosidic bonds and use the sugars in carbs for energy. And so these would be reaction energy diagrams of making the carbohydrates and then using the carbohydrates. Okay. So another name for carbohydrates is saccharides. Right, so this is another name for sugars / carbs. And monosaccharides are the simplest unit of a carbohydrate. So this is a single carbohydrate, like the ones we were looking at, identifying before. Those would be classified as monosaccharides because it's a single carbohydrate molecule. We can have multiple monosaccharides that are linked together with glycosidic bonds. So these are linked together with glycosidic bonds. And so these become usually other forms of sugars or carbohydrates. And we have multiples linked together, so they're called mono. Then we have disaccharides, trisaccharides, and then anything after that we usually just call a polysaccharide. And this is usually when we consume complex carbs. Mono and disaccharides tend to be sugars and stuff that we use. So sucrose, for example, is a disaccharide of glucose and fructose linked together. And so this is kind of like the hierarchy of carbohydrate structure. We have polysaccharides, we can break those into tri or disaccharides, and then those are composed of monosaccharides, which are the simplest unit of carbohydrates. In most biomolecules we're looking at, usually there's one simple unit that then makes a polymer to generate the more complicated full molecule. Okay, and so how do we classify monosaccharides? So we classify them based off the functional group. We have a suffix of -ose. So you'll notice most carbohydrates have an -ose ending. That lets us know that this is a saccharide or a carbohydrate. Saccharide or carb. And then we classify them based off of the aldehyde functional group. or the ketone functional group. Aldehyde functional groups are called aldoses. Ketone functional groups are called ketoses. And then we have a prefix to indicate the number of carbons in the carbohydrate. And so glucose and fructose are specific examples. And so I'm not going to have you guys remember the specific names for all these carbohydrates. More so look at the structure and determine things by looking at the structure. So glucose and fructose, I don't need you to memorize that structure, but if I tell you this is glucose, classify what kind of carbohydrate it is, you should be able to say that glucose is going to be an aldohexose, and fructose is going to be a ketohexose. Aldo tells us what about this carbohydrate? An aldehyde functional group. Ose tells us what? It is a carbohydrate or it is a saccharide, right? And then what do you think hex tells us? This is going to be the number of carbons, right? Number of carbons. Keto for fructose tells us it's a ketone. Hex tells us number of carbons. And then ose tells us that it is a saccharide or a carbohydrate. And so I know we have a lot of prefixes we've already talked about. So carbohydrates go back to the prefixes that we used for covalent compounds. So I'll list those in a second. But what do you guys think is going to be the smallest prefix we will use for a carbohydrate? How many? 3 carbons, right? So tri is going to be the smallest prefix for these. So an aldotriose would be the smallest carbohydrate classification you'll have. And so the prefixes are going to be for the number of carbons, and then the prefix, if we have 3, that's tri, 4 is going to be tetra, 5 is pent, 6 hex, 7 hept, 8 oct, and then 9, no, but I'm never going to give you a 9 carbon carbohydrate. And the general structure for the name is going to be the functional group, then the prefix, followed by ose. So it's always going to start with aldo or keto, then the prefix is the only thing that's going to vary from each of the classifications here. Carbohydrates. Yeah. Oh, yeah. Those are all this is all for carbohydrates in general. Once you get to poly and disaccharides, you don't classify it as an aldo or ketohexose anymore, because if you have something like sucrose, which is a aldohexose and a ketohexose bonded together, those have two different functional groups in there. So those are just complex. Those become complex carbohydrates at that point. Really, this is for looking at an individual carbohydrate. For monosaccharides is what we do this type of classification for. And so how do we represent carbohydrates? I've drawn this a lot so far, but so we represent them with what's called a Fisher projection. And what a Fisher projection does is usually when we draw organic molecules, we have them bent in a structure like this. And then we have stuff that comes in and out of the page. And so when we talked about organic chemistry, we talked about the idea of chirality. Does anyone remember what chirality was? These are specific type of isomer called stereoisomers and enantiomers. And chirality is when we have two non-superimposable mirror images, right? Like your left and right hand, they're a mirror image of each other, but there's no way you can superimpose them on each other, right? Which means the structure and the bonding, three-dimensional structure of this is going to be different, right? So these were non-superimposable mirror images. And so these were also sometimes called optical isomers, because if you have chirality in your molecule, it rotates light differently. And for chirality to exist in a molecule, you need an sp3 carbon. And sp3 carbon was what shape? Tetrahedral, right? You need a tetrahedral carbon, and you need 4 different groups bonded to it. And when you have this specific set of circumstances, these carbons are called chiral centers, meaning that those carbons give a molecule some level of chirality. And so with carbohydrates, every single carbon in the chain, except for the very last one and the very first one, if it's a ketone, are going to be chiral centers, right? Because one group is a hydroxyl group, one group is a hydrogen, and then it's asymmetric because if you go along one side of the chain, you get to the functional group, aldehyde or ketone eventually, and then you go the other way, you'll get to your final carbon in the chain. Those groups themselves have different chemical formulas, so you have 4 different groups attached to it. And so what we do when we represent these carbohydrates is we take this 3-dimensional structure, right? So the bolded blue lines there, that means those groups are coming out of the page. The dashed ones mean that they're going into the page, right? So I'm trying to represent a 3D structure in 2D. And the way you draw a Fisher projection is that this becomes a flat line And you kind of, if I'm looking from the top of this, I smush it down so that everything gets sprawled out in a flat way. So this would look something like this. I've taken this three-dimensional structure and now I've represented it in a simple 2D structure. Here, if we had 2 hydrogens or 3 hydrogens, they would be drawn like this. And these would not be chiral centers because we have more than one of the same group. Right, where the blue and the red are, those are going to be chiral centers. And so these different specific type of isomers that have these chiral centers we've talked about before, these are called enantiomers. And so this is a basic idea of how we generate a Fisher projection. Because we're trying to get this three-dimensional structure from a 2D representation. And ultimately, what we're going to end up doing is we're going to align the whole structure such that, remember I talked about that penultimate carbon before? When we flatten that structure according to the penultimate carbon in a specific orientation, it'll fix all of the other chiral centers into another position. Right? So we only have to worry about one carbon in terms of which enantiomer we're dealing with. It's a lot easier than looking at each carbon in the chain and determining what enantiomer we're dealing with. And so in the notes that I posted with the completed version, I went into a bit more detail on how to generate a Fisher projection. I'm not going to talk about that. Really, the important thing is being able to look at a Fisher projection and understand what it is that you're going to be looking at. And so I'm going to skip ahead to the specific enantiomers we have in carbohydrates, which are D and L isomers. And so the D or L isomer is determined by the direction of the hydroxyl group in the Fisher projection on the penultimate carbon. L enantiomers, we can remember this, L enantiomers have the hydroxyl group to the left. Right, so L enantiomers have the hydroxyl group to the left. Here I put R on accident, but this is supposed to be D. And D enantiomers just have the opposite. They have the hydroxyl group to the right. And so if I draw, I'll probably do this on a different slide, or I'll go to the next slide actually. And so the only thing that matters if we have a D or an L isomer is going to be which direction the penultimate carbon's hydroxyl group faces. And so does anyone know in nature which preference there is from biology class? In nature, we only have D carbohydrates, right? All natural occurring carbohydrates and sugars are going to be D isomers. Nature, let me put D carbohydrates or saccharides are the only ones that occur naturally. I put 2 T. so what does an example of this look like? Well, Fisher projection is always going to be vertical. So a Fisher projection is always vertical with the aldehyde or ketone at the top. We have a straight line. If I draw this at the top, how do we classify this carbohydrate? It's an aldose, right? We don't know what classification is beyond that yet. At the end, we're always going to have this CH2OH group there. This carbon, does anyone remember what that was called? It's the carbonyl carbon, which in carbohydrates is called the anomeric carbon. And then we're going to have a bunch of hydroxy groups on the carbohydrate, so I'm just making this one up. And on the other side, we're going to have hydrogens, right? So if we look here, we see that all of these carbons right here are sp3, and they have 4 different groups on them. So they're all going to be chiral centers. But the only thing that determines if it's a D or an L isomer is going to be the penultimate carbon, right? Because here, with this carbon, we have 1 hydrogen, 1 OH, then we have this whole thing down here, and then above it, all we have is the aldehyde, right? So there's 4 different groups. It's the same thing if we analyze the second carbon after the aldehyde. We have 1 OH, 1 hydrogen. Now we have this group down here, and now we have 1 hydroxyl group carbon and 1 aldehyde above it, right? So these are the 4 different groups that are attached to this chiral center. We don't worry about all of them. We just worry about the penultimate carbon. So which one was the penultimate carbon? Second to last, right? So this is the penultimate carbon, right? The intersection represents the carbon atom there. Remember, we actually have a zigzagging chain because they're all tetrahedral carbons in this chain. But what we've done is we've oriented the last group and we smushed the structure so that it sprawls all the bonds out, and that's how we're representing this structure here in the Fisher projection. And so if I ask you, is this going to be D or L isomer or enantiomer? Which one is it going to be? The only thing we have to worry about? It had to do with the penultimate carbon. And what did it have to do with the penultimate carbon? The direction of the hydroxyl group, right? If it was L, the hydroxyl group was on which side? The left. And if it was D, it was on the right side, right? So which direction is the hydroxyl group here? Left. So this is going to be in L enantiomer. OH on left, L enantiomer. So if we were to fully classify this carbohydrate, how would we classify it? First, we would put that it is L. And then how would you classify the rest of this carbohydrate based off the other rules and prefixes we just said? We put aldo, right, because there is an aldehyde functional group. How many carbons are in this chain? We have 1, 2, 3, 4, 5. So what would the prefix be? Pent. And the ending for a saccharide or carbohydrate would be ose, right? So this would be an L-aldo pentose.
So was that in
Yep, that's because, right, so remember when we name aldehydes and ketones, if it's the highest priority functional group and it's higher than alcohols are, right? We always start numbering from that part.
One's 2 carbons?
It's only 1 carbon there, right? So this counts as only 1 carbon. That's a good question. This is 1 carbon that's double bonded to the O, right? That's our carbonyl. This, if we were to draw it in a carbon skeleton, would look like that. Does that make sense? And why isn't the anomeric carbon a chiral center?
There's no mirror image.
There's no way you can not generate a mirror image, but it has to do with one specific property of this carbon, the hybridization of it. It needs to be sp3, right? And what's the hybridization on a carbonyl carbon? How many bonding groups does it have? 3. So it's going to be sp2 or trigonal planar, right? So that's another reason why you can't. And whenever you have a trigonal planar, the reason you can always generate a mirror image of them is because you can always flip it around, right? You can always flip it around that plane and generate a mirror image. You can superimpose a mirror image, right? So if I, just for illustration, if I have something like this and I write a mirror image, I can always take this and rotate it 180 degrees, and now the mirror images are superimposed on each other. So they're really just the same molecule. But yeah, so that's how we would classify this according to its enantiomer. So here we have 2 different carbohydrates, very, very simple carbohydrates. These are both, how would you classify them in terms of their functional group? In terms of their functional group, so not the enantiomer yet. They'd be aldoses, right? And they would be aldo, what would be the prefix for both of these? Aldo, that's the suffix. 3. Triose, right? These would both be aldo trioses, right? Because of the aldehyde, and they have 3 carbons. And so on the left one, is that going to be a D or an L isomer? D, right? Where is the penultimate carbon? What number carbon is the penultimate carbon? Yep, the second one, that's the penultimate carbon because it's always the second to last carbon. And this is going to be D because the OH group is on the right. And what about the other aldose here? It'll be L. This is the penultimate carbon. It's on the left, which means that it's an L enantiomer. So actually, once we get into the ring structure, these molecules, you can't do it. You need to have a pentose or a hexose. Let's say we have a aldo hexose like this. This CH2OH group, which is the last one in the chain right there, either goes above the ring or below the ring. And so depending on where that CH2OH group is, it forms a different type of bond, right? So this will restrict the orientation if you form an ester bond on the top or an ether bond on the top. We'll talk more about that. And it's not going to be super relevant for the class, but an ether bond on the top or an ether bond on the bottom, that'll mean these rings will polymerize in different shapes. So that's why it's important for the ring structure. That's the consequence of D or L isomers once we get to the ring structure. We'll talk about that probably after a break, though. All right, any other questions on enantiomers here? And the fun thing is once we get to ring structures, there's a different type of enantiomer that has to do with the hydroxyl group here, and we'll have alpha and beta anomers instead of enantiomers. All right, so let's take another 10-minute break. We'll come back and hopefully finish up carbohydrates. This one?
Yeah.
This one or the next one?
Yeah.
That one? Okay, yeah, I'll leave it right here. this one. So these are the D or L isomers. So here are some important hexoses that you see. We have fructose, galactose, which is the carbohydrate milk, and glucose, which is a sugar. So for each of these, what is the enantiomer for each of these important carbohydrates? Which enantiomer is each of these? They're all going to be D, because the hydroxyl group on the penultimate carbon is on the right. So these are all D enantiomers. And this makes sense because all naturally occurring carbohydrates are going to be D enantiomers. Like I said, I'm not going to have you memorize what the specific enantiomer is or what the specific structure of glucose is, but look at this and I'll tell you it's glucose and then classify it based off of that, right? So we see galactose and glucose are both what kind of carbohydrates? Like how would you classify them? They're aldoses, right? And they're aldohexoses, right? Because they both have 6 carbons in their chain, right? So both of these are going to be aldohexoses. And then fructose is what? A ketohexose, right? This is going to be a ketohexose, right? Because this is a ketone, and the other two are aldehydes.
How is the glucose and galactose?
The orientation of all the other hydroxyl groups are different from each other, right? So we look at this. Yeah, you see this hydroxyl group goes to the right, that one goes to the left. So this is why you only have to worry about the penultimate carbon, because when you smush this structure down, it fixes the location of all the other hydroxyl groups. So these hydroxyl groups are in this position relative to each other. And that's what gives it the structure. Once you go to the ring structure, it means the hydroxyl group is going above or below the ring. Okay, I'm good to go to the next? Not yet. And then the plus or the minus sign you don't have to worry about. So this is a specific optical property of these, even though these are both D, or all these are D isomers. The plus sign means that it rotates light clockwise, and the minus sign means it rotates light counterclockwise. And so it's not important, but that's why those plus and minuses are there. So a beam of light hits this, it rotates the light in a different direction. And so we can have these cyclic structures of monosaccharides. And so if we have 5 or more carbons, we can form ring structures. And what happens is when we have aldehydes or ketones, they can react with alcohols to form what are called hemiacetals. And this gives a ring structure for saccharides, because what you're having is you have one alcohol side of the molecule reacting with a ketone or aldehyde side on the other side of the molecule. And so if you have a ketone or an aldehyde, this will react with an alcohol, usually in the presence of water, to then generate this structure which is called a hemiacetal. Or this would be called a full acetal, but it can also be, this is a hemiacetal because we have one side. We have an ether that is next to a hydroxyl group. And I wouldn't stress about this reaction. This isn't a reaction that I'm going to give you guys to predict the products for. This is just to give you an understanding of how we form these ring structures. And so this could be a ketone or an aldehyde. If it's an aldehyde, it just means one of these R groups is going to be a hydrogen or replaced by a hydrogen. And so this happens spontaneously in the presence of water. So in solution of saccharides, they're going in between their cyclic and their linear structures relatively frequently. Depending on solution conditions, you can push them one way or the other. No, go ahead. I'm curious now. And so here, these are between two different molecules, an alcohol and a ketone. But if we look at our carbohydrates, they have one side, that's an aldehyde or a ketone, and the other side, or on the rest of the molecule, we have alcohol groups. We have the hydroxyl groups, right? So this means we can have what's called an molecular reaction, where we have this reaction not between two different molecules, but two different parts of the same molecule. And so this is going to close the structure and form a ring. And so what this is going to look like, or how we get to a ring structure, is let's say we have a simple 1, 2, 3, 4 CH2OH, H, H, OH, OH. We have a D, and then this is going to have how many carbons in it? It should be 1, 2, 3, 4, 5, right? So it's an aldopentose because of the aldehyde functional group, and a pentose because there's 5 carbons. And the way we go to a ring structure from this is we rotate this 90 degrees. So this is going to look like HO, H2C. Let me make this a different color, sorry. HO, H2C. Our aldehyde, and we have 1, 2, 3. We keep the orientation of the groups the same. H. And everything that's not the anomeric carbon or the penultimate carbon is going to keep this orientation when we moved it to the side in its associated ring structure, right? So this is the penultimate carbon. Because this is what told us is if it was D or L. This is the anomeric carbon. And so the only two spots that are going to keep their orientation on the ring are these two carbon groups, right? So these are going to be the same on the ring structure. And so what happens is, and this isn't gonna be something that you're tested on, is that this aldehyde part of the functional group reacts with the alcohol side of the penultimate carbon, right? And so if these two come together, they're gonna form a hemiacetal. But they're part of the same molecule, which means that all of these carbons in between are going to be a part of this loop that's been created. And so for an aldopentose, you're going to create a 5-member ring. One spot on the ring is going to be taken by an oxygen, and then the other spots are going to be taken by carbons. So it's a 5-member ring, so it'll look something like this. The first position on the ring, we either have an OH going up or down, and we'll talk about what that means. There's no way that you would be able to predict this beforehand. It would be something that has to be given to you. And this carbon in the ring is the anomeric carbon. This carbon in the ring is the penultimate carbon. And so if I was to number them on the original ring here, this was 1, 2, 3, 4, 5. The anomeric carbon was which number? 1, right? Because this is the anomeric carbon, right? Which is the carbonyl carbon. 1, 2, 3, 4, 5. This is the anomeric carbon, so the ring is going to start numbering from here. So it's going to be 1, 2, 3, 4, and then you say, okay, wait, we need another carbon here. Where does it go? Well, remember that this reaction is happening between this oxygen here on the penultimate carbon and the carbonyl carbon, right? So that's where they're being closed. So what happens is, if this forms the loop here, this is off to the side, right? So here, the loop is being formed here. That's going to be our ring. This is the oxygen that's linked to the anomeric carbon. And so this CH2OH group is off to the side. It's been excluded from the ring, right? There's no way we include that in the ring. And so the CH2OH group, which is our last carbon, is either going to be going up or down on this 4th carbon. If it is D, the CH2OH goes up. So this is for a D enantiomer. If it is an L enantiomer, it will go down, right? So it's hard to draw that, but basically this ring is coming in and out of the page right here, and CH2 for a D enantiomer is going up, and CH2OH is going down, right? In every spot that we don't have a CH2OH or an H, the opposite is going to be hydrogen for all of these. So really what gives us the orientation of the entire molecule is the orientation of these hydroxyl groups. And so all we do with the middle spots, right, if we look at the ring here, we have these going up and down. Those are the 2 spots that are in the center of our Fisher projection. And when we rotate it to the side like this, you just keep the same orientation and add the hydroxyl groups and hydrogens based off of what they look like in this laying down structure here. Right, so I'm going to number this structure on the side just so we can keep track of this. This is 1, 2, 3, 4, 5, right? This was 2 here, 3, 4, then 5 is off the ring, so we're not worried about that. So on the second carbon, the hydroxyl group should go which direction? Up or down on the ring? Right, the OH group is pointing up or down here? It's up, so when we weight it on its side, we keep this the same orientation as we do on the ring. So the OH group is gonna go here, and here, what are we gonna have? Hydrogen. Right, we have OH and then H down. So this is going to be OH and then H. And what about on the third carbon? What's going up and what's going down? H is up and OH is down. Right, so what we've done is we've taken, you can imagine I grabbed this oxygen here, and I grabbed the anomeric carbon, and I bent the ring to form a ring, a bond between those two. When I did that, I ignored the CH2OH, which is why it's not a part of the ring. So this isn't a part of the ring because it's been ignored from the formation here. And then I rotate the structure on its side, and the orientation of all of the groups that are not the anomeric carbon or the penultimate carbon stay the same, right? So this is why we rotate on its side. This is just a trick so that we know how to put all the hydroxyl groups on the carbohydrate in the ring structure. And so this is how you actually get different structures based off of the type of carbohydrate when it becomes a polysaccharide, because these OH groups can react with other carbohydrates to form what we call a glycosidic bond between them. And so if you have one in a hydroxyl group going up or down, that determines the shape of whatever your polysaccharide is going to be.
Would be like D or L?
Yep, and the D or L, right? So remember you asked about that before. If you react with this hydroxyl group versus in the L structure, the next chain, or the next is going to go down. Yeah, the next one is going to go down, whereas this one, the group is going to go up. So it changes the orientation of the bond it forms with other saccharides. So that's the difference between D and L. If we have a ketohexose, what do you guys think will change here, if you had to guess? All right, so this is an aldose, right? Right, so the anomeric carbon, we have what other group bonded to it? We have this, and then what else? Hydrogen, right? And this hydrogen has been excluded from the ring, right? Because we're taking this carbon and bonding it to this penultimate hydroxyl group. If we have an extra CH2OH group, we do, in a ketose, where do you think that's gonna go on our ring structure? It's gonna go off the ring right here, right? Because it's being excluded the same way this is. So the distinction between a ketose is it would have to be a 6, so this would be a D-ketose, CH2OH, is that we have the OH group going up, that's this carbonyl carbon, this carbonyl oxygen here, that gets returned into an OH group, you don't have to worry about that. But the opposite side is going to be a CH2OH. So we have 2 CH2OH groups instead of just the one we do with aldoses. And I didn't draw all the other groups here, but all those other groups would be there as well for both of these. And that's because if you erase this hydrogen and you add CH2OH there, you're still taking this carbonyl carbon and pulling it to that oxygen here and closing the ring. The CH2OH group is excluded from the ring the same way that this one is. And so it's hard to see based off the reaction that I showed on the previous page, but this is your hemiacetal, because this is one R group over here. This part of the ring is another R group, and then this is your ester part, and then your hydroxyl. right? So the important part, the takeaway from this isn't exactly understanding all the chemistry that's happening here, but just if we have a linear Fisher projection, you can draw the ring structure just by rotating it on its side. If it's D or L, the CH2OH group on the fourth carbon in the ring goes up or down. And then if it's an aldose or a ketose, instead of the H on the anomeric carbon, you have a CH2OH. Right, so those are the important distinctions here. Everything else, doesn't matter what type of carbohydrate it is, all of the intermediate groups here are gonna stay on the same orientation when you lay it down on its side. All right, you guys want an example to see if you can figure out how to do this? I'm not gonna do glucose here. I'm just going to draw to OH 1, 2, 3, 4, 5, 6. See if you guys can draw the ring structure. So there's a couple things we need to identify right away. Is it a D or an L enantiomer? Is it an aldose or a ketose? Those are the important things. Then once you have that, count the carbons in between. So 1, 2, 3, 4 carbons are going to be in the ring, and then the O is also going to be in the ring. So it'll be a 5-member ring, right? Because one spot is always taken up by oxygen in the hemiacetal part of the ring. So then draw a 5-member ring and see if you can fill in all the spots. All right, to do that, you're going to rotate this on its side. And then the D enantiomer tells you where the CH2OH group is going to go. Aldose or ketose tells you if you're going to have an extra CH2OH group or not. The orientation of the OH or CH2OH group, you don't have to worry about, because I'll have to give that to you. You actually get exchange between the two. So first thing, is it an aldose or a ketose? Ketose, right? Because we have the extra CH2OH up here. This is going to be a ketose. And is it D or L? It's on the left, so this should be, this is the penultimate carbon, so it's going to be an L in an enantiomer. I already counted out the carbons. We have 1, 2, 3, 4, 5, 6, right? But the bond is going to be between the anomeric, so it's going to be 1, 2, 3, 4, and the penultimate carbon, oxygen. So we're going to have a 5-member ring. So I'm going to draw the ring. I'm always going to draw the oxygen first, and then draw the 5 carbons around it. It's going to look something like this. I'll try and draw the orientation a little bit better. The OH group, I'm going to put up right now because we haven't talked about what that means, and I have to give that to you. But we can fill in some other things right away. So because it's D, what can we, an L enantiomer, what can we put in right away? Yep, the CH2OH on, and let's number the carbons. So this is going to be the second carbon we have there. This is 3, 4, and then 5 here. So where are we going to put a CH2OH group? Yep, we're going to put 1 on 5, and that's according to the D or the L isomer, right? So this is either going to go up or down. I'm just going to draw it off to the side because it's a little hard to draw going down here. And so is it going to go up or down? Down because it's an L isomer, right? So H2 or HO, H2C. This is because it's an L enantiomer. Sorry, I keep saying isomer. Where else are we going to have a CH2OH group? On 2, right? And that's because why? Yep, it's because it's the anomeric carbon, and the anomeric carbon in a ketose doesn't have a hydrogen there bonded to the other spot. It's a CH2OH group. This is because it's a ketose. And then I'm just going to draw lines for all of the other positions on the ring, right? So this was the hardest part. Once you have this, then we can just take this structure. See if I can rotate it or figure out how to rotate it. Duplicate. I can take this structure, rotate it 90 degrees, and then I'm going to circle everything in between the penultimate carbon and the anomeric carbon, and I fill in the spots according to their orientation right now. So on the third carbon, what goes up and what goes down? Yes, I guess the orientation is a little hard, so you gotta kind of read sideways because I just copied it and rotated it, right? But what goes up? The hydroxyl, right? So the hydroxyl group is going to go up, and then down, we always have a hydrogen in the other spot. On the fourth carbon, the hydroxyl group is going to go down, and the H is going to go up. Right? And that is our ring structure for this arbitrary ketohexose that I drew. And then this other position is also going to be a hydrogen that goes there. And so generally, if I ask you something about this, I will give you the ring, I'll give you the position of the hydroxyl group, and I'll say fill in the other spots. So the stepwise thing is determine if it's D or L, determine if it's a ketose or an aldose. That tells you the position of these two groups and the identity of those two groups. Then you just rotate it on its side, or if you're good at visualizing it, you can just look at it sideways, and that gives you the orientation of the polyhydroxyl part of the carbohydrate. Alright, so I know it's a lot of steps for this, but this is the, you know, how the actual structure of the molecules come into play, because this is how carbohydrates are going to polymerize with each other. They don't polymerize in this linear form, they polymerize in the ring structure. Okay. Say that again? Here, so this, third carbon, OH is up, OH is up. Hydrogen down, hydrogen down. Oh, I said this is the polyhydroxyl part, right? Because this is the multiple hydroxyl groups on each of the carbon atoms here, the multiple hydroxyl groups. Any other questions on this? Okay, and so the last thing that I'll talk about before we end, so I'll probably finish carbohydrates on Thursday and then start proteins, is the stereochemistry of the ring structures. And so depending on the type of carbohydrate, the aldose or ketose, different anomers have different stereochemistry. And so these are a specific type of enantiomer called anomers. And on the first or second carbon, if it's an aldose or a ketose, the direction of the hydroxyl group gives us the anomer. The direction of hydroxyl group gives the anomer, or the specific anomer. And so if I draw an arbitrary ring structure, we'll do aldohexoses. CH2OH, H, H, OH, OH, H. I'll duplicate the structure. Duplicate. Duplicate. So we have two different anomers of each other. If the OH group is up or the OH group is down. This is a beta anomer and this is an alpha anomer. All right, so the only thing that tells us the type of anomer is just the orientation of this hydroxyl group. And so this is why the carbonyl carbon is called the anomeric carbon, because that double bonded O is what gives us the anomer. That double bonded O turns into this hydroxyl group, and it goes through a chemical reaction to then reduce this into a hydroxyl group. And it doesn't matter what aldose or ketose you have. It doesn't matter if that H is a CH2OH or an H. All that matters is the direction of the hydroxyl group on the anomeric carbon. And so the last thing I'll mention about anomers, well, the last thing I'll mention today is you can get an exchange between these anomers in an aqueous solution. So if you have water around, you can exchange between the alpha and beta anomers. And so what that means is it's called mutorotation. And so you can exchange between anomers in aqueous solutions, so just water solutions. And the anomer is important because this defines the type of glycosidic bond that will link saccharides together to form polysaccharides. You will either have a alpha 1,4 glycosidic bond or a beta 1,4 glycosidic bonds. And that's important because those different bonds store different energies, which means you get different calories from eating them. And so this is the exchange between alpha and beta. I could draw the structure, but I know it takes everyone a long time to draw the structure. I've done it thousands of times now, so I can get kind of quick at this, but I know when you're drawing these structures for the first time. Trust me, if you guys go on into organic chemistry, you'll get really good at drawing hexagons, pentagons, octagons, and all of that.